Hello, I'm back again. The time is going so incredibly quickly, like having any sort of time in between these live streams. It's ridiculous. Welcome back for another chat with Going Live with Global Science Show. We're here for another 15 minutes to talk to another science communicator about who they are, what they do, and why they're awesome. And our next guest has taken part in Going Live with the Global Science Show before, but clearly can't get enough, is back to have another chat. Again, you can join in the conversation if you like, using the comments on Periscope or on YouTube, depending on where you're watching. Please welcome into the stream, it's Alex Holmes. Hello.
Hello, thank you for having me back.
No, I'm delighted to have you here. For those who don't know you, who are you and what do you do?
So I'm Alex Holmes. I am a PhD researcher at the University of Leeds, and I study biochemistry and biophysics of membrane proteins. So it doesn't look too closely linked to biodiversity for today's show, but I just did a placement with the BBSRC where I was working on genomics. So I do have a little bit of an inkling about biodiversity and genomic sequencing now.
So you could be seen as one of those like imposters, like why are you here to talk about this? But there's
Yeah,
actually a reason.
100%. Don't kick me off the
Okay.
show. I'm sorry for admitting that immediately.
No, I promise that I do have that power of God, but I'll keep you here as long as you play nice.
I will.
So earlier on, you and Sam talked about, other Sam Rowe talked about genomic sequencing, and he talked about the Darwin Tree of Life project. So it'd be really good to talk about like what uses does genomic sequencing have as a tool for studying animals. Too many live streams.
That was like alliteration central.
Yeah, so I'll try it again. What uses does genomic sequencing have as a tool for studying animals?
Perfect. So I think when you're a scientist, your main kind of trait is just curiosity. And I think it's so true that scientists in the past were looking at animals and going, what is that? And what makes it like that? And if you know about genetics, basically your genetic sequence links up to kind of who you are as a person, what your traits are. And so being able to sequence the genome of an animal means that you can study its traits, understand what's going on with it. But years and years and years ago, kind of the 80s, so way back in the dark ages, you couldn't sequence genomes. So you'd have like entire PhDs, like 4 or 5 years, just trying to understand a single gene out of maybe 30,000 genes in an animal. So being able to sequence all the genes at once and then being able to work with them just massively accelerated things and let people kind of pick up a whole animal and go, what is this? What is it doing? Rather than going, oh, we've got a whole animal, but I'm going to zoom into this like tiny little piece of DNA and try and work out what that's doing.
I think you've just insulted a whole bunch of people that might be watching this by like referring to the 80s as like the dark ages and the before times, and yeah,
I know.
it'd be great to talk about model organisms, because that comes into a lot of what we're discussing today. For those who don't know, could you tell us what a model organism is?
Yeah, sure. So model organism is basically an animal or other kind of life form that we can do science in and study as a kind of proxy, approximate for a human. So humans would be an absolute nightmare to study in a lab. You can't just breed them together willy-nilly to see what happens. You can't keep them living there for kind of 80 years to study what they do in old age, because you too would then also be in old age. And generally they'd just be really difficult to kind of keep in a lab. They're quite big, they're quite noisy, they like to have a lot of entertainment. So what scientists do is they kind of pick animals that have similar properties or traits so they can then study things in, so they don't have to house humans all the time. And it's a bit more ethically sensible to kind of keep rabbits, guinea pigs, flies, rather than keeping like a whole family in your lab.
So 2 points, right? You know where my brain's going is that you mentioned we couldn't keep humans in a lab until they're 80 because you too would be old as well. And my brain, and it's the coffee, went Bono and the Edge, really, really old. Like U2, I'm just like really, really old members of U2 in the lab. That is where my brain went. And I'm not sure why. But
Yeah, all
obviously
the pop
we
stars, when they retire, that's where they go, straight into the labs.
go live in the lab. But obviously we couldn't ethically keep Bono in a lab as much as some people might want to do that. How do we ethically use model organisms?
That's a really good question. So there's obviously a lot of debate about animal experimentation, and it is something that's unfortunately still necessary, just because we can't study humans and we don't have kind of other options that are up to scratch. But there's an awful lot of regulation. So anywhere that's using animals has to have a Home Office license. They get regular checks and random checks to make sure everything's up to scratch. And there's also these kind of 3 guiding principles called the 3 Rs. And that always makes me think of like reuse, reduce, recycle, but not quite. It's still reduce. So if you can get away with using less animals for a particular study, that's recommended. If you can refine the study so that you can use a different kind of animal that maybe works better for the particular question you're asking, or maybe doesn't, I don't know, feel pain in the same ways, or is not kind of as higher order as other organisms are. And also the final one is replace. So that is where you then could use a cell line instead of using animals or something like that.
Okay, no, that makes sense. But I imagine it's quite a big discussion that a lot of people have some real strong opinions on how we use animals within research and within labs, and knowing that kind of guidelines and the licenses you need to have really just show how strict it is in terms of how you
Yeah,
use them.
and it's stricter than having pets. So for example, I have a hamster, and when I got her, kind of, they just basically gave her to me, and I had all the stuff to make sure that she had a lovely life, but no one's going to come check on me every 6 months to make sure she's still in a cage that's big enough. No one's going to come check she has the right bedding, that she's happy. Whereas animals living in research labs, they have huge enclosures. They're constantly be giving like nice food, things to play with, all sorts of things. Like, and if I was found to be mistreating my hamster, which I'm definitely not, I probably wouldn't even be arrested. I might just be fined. Whereas if I was found to be mistreating a lab animal, I could be arrested, and like the entire department could have kind of inquests and things. So it's really tightly regulated.
It's a really good kind of comparison to make between like how pets there's so unless it's like a dog or a cat, especially dogs, there's much less kind of restrictions there on who can get one and what you get to do with it.
Yeah,
Hmm.
exactly.
So, what sorts of new avenues has genomic sequencing opened up for research with animals or like not having to use animals?
That's a really good question. So one of the things I was looking at when I was at the BBSRC was sequencing of new animals, so things that we hadn't sequenced before, didn't have much information on. And one of the like main examples, so I was going to have another main example actually about flies and other kind of insects. But actually, one I think ties really nicely into refining animal experiments is fish. So a lot of people use zebrafish in experiments. Their embryos are see-through, so you can see them developing and all their kind of bones growing and things. So they're really cool to study. There's recently been quite a lot of research into alternatives to zebrafish, like something called killifish, and they might have slightly different reactions to analgesia and yeah. What's the thing that puts you to sleep? It's been a long
Anesthetic.
day. Anesthetic, thank you. Thank you, Sam. Um, so there's probably some circumstances where we could use killifish over zebrafish because they would respond better to anesthesia, and it would just be better to use them. But also then with the fly example I was going to use, we studied a lot of things in Drosophila for a really long time. So this is kind of a normal fruit fly that you can live in the lab. And there's more and more sequencing of other insects going on. So, for example, there's a wasp called Nasonia, which is parasitic. And this actually, you can study something called epigenetics in it, which you can't do so easily in Drosophila. And there's all sorts of other insects that are being sequenced. So, for example, crickets, and trying to understand how crickets hear, because crickets have to make noises to communicate with each other. And obviously, humans do the same, but flies don't do that. So we can study hearing in crickets and how they process sound. Or there's butterflies and how their wings evolve to look like other animals, so kind of mimicry, which humans don't do. But it's really cool that we can study that in butterflies and understand how is it that this wing also looks like tree bark, or this wing looks like a face, or whatever it is.
Hmm. Well, that's great. I really like that.
It's really cool.
So, like, there's so many different types of like model organisms and ways that we use them. Actually, you mentioned epigenetics. For those who are watching who might not know what that is, what is epigenetics?
That's a really good question. So if you think about your genome, which is kind of the set sequence of A's, T's, G's, and C's, those letters, all in order, it's all bundled up really tightly in your nucleus of the cells, and it's really small. So only certain bits can be accessed at a time, and bits get turned on and off. Or actually, sometimes things can be dimmed and undimmed, a bit like a dimmer switch in your house. And epigenetics is kind of studying which genes are turned on at certain times and how this kind of changes throughout your life. I did have a really good example of this, but it has escaped me at the moment.
That's fine. Like it's always when you want, like, it's always when you're live and you want a really good example, it disappears. But that's a really good introduction to what it's all about. So why can't we study epigenetics in fruit flies, Drosophila? Is it to do with the length of the genome being shorter,
Yeah,
or
length the genome, and also they just don't need epigenetics in the same
okay?
way. So they don't need to kind of adapt within their own lives to kind of change to different environments and things. So actually, one of the examples actually that I do have there in my mind is kind of fetal development. So early on, when you're a fetus, little baby, you need to have certain genes turned on so that you can develop and grow quickly. When you get older, these get turned off and you stop growing. But this is because we have a much longer life cycle. Whereas if you're a tiny little fruit fly, you don't really need this in quite the same ways.
Hmm. Okay. Well,
And I
that
think,
makes sense.
yeah, I think fruit flies are always, they're also really easy to kind of genetically modify. And it's quite cool to then try and do this in other insects that maybe aren't so easy to do this in, because you can study completely different things and different regulatory patterns of gene expression and things.
Hmm.
That sounded really fancy when I said that.
You sounded like you knew what you were talking about, like, which
Impressive.
is, yeah, like that's half the battle. Sometimes it's sounding like you know what you're talking about. We're getting towards the end of the 15 minutes, but there's one or two other things I wanted to ask. The first one is for people who are watching who have maybe got no sort of connection to genomic sequencing or anything to do with model organisms as well. Why should they care about the work that we're doing to sequence the biodiversity around us?
That is such a good question. So I think if you think about things from that really like sciencey perspective of there's only so much you can study in a lab and there's only so much you can study within your own lifetime, really. Whereas evolution has been going on for millions, billions of years, and it's kind of tried out every variant it can do and played with genomes as much as it can, and probably will continue to in future. So when you're thinking about, oh, it'd be really cool if we could design an animal that might do X, Y, Z, chances are evolution's already tried that out and found the most efficient way of doing it. So by studying and sequencing a lot of animals, you can really understand all these different things evolution has tried and not have to spend a lot of time or reinventing the wheel or reinventing the fly or whatever it might be.
Great. And that kind of brings you to that idea. Like it's always something that I like think about when it comes to well, evolution's tried that is the fact that crabs have independently evolved like 4 times separately or something. Like evolution really likes crabs.
Understandable, they're very cool.
Yeah.
And like
Yeah.
even crabs are really cool to study because they have like compared to humans, completely different like time cycle. Like we're on 24-hour clock, they're on 12.4 because of the tide cycles. Crabs molt, we don't molt our shells, we don't even have shells. Like, yeah, you can just see all these things that you wouldn't see anywhere else in the world or in biodiversity because they're being done by something else entirely.
Oh, amazing. It's been a really great chat and we've ran over time, which shows that it's been great. Thank you so much for coming to chat with us.
No, thanks for having me on. I've had a great time.
Lovely. I'm going to use my power, as always, to kick you out of the room. But thanks, Alex.
Thank you.
another one done. We've just we should have just had another post quote out from M. Dixon as well, which I'm really, really excited to see what that's all about, because it's usually a bit mind blowing. So yeah, please go check that out at Not So Sci-Fi. We'll be back again for another live stream in 45 minutes time, and we're going to be talking about some microscopic marine worms, which is going to be really great. And then at 2 o'clock we'll be back again, and I'm going to have some extra guests who are going to come on to the live stream as I try and figure out how to set up some extra cameras to have a look in some animal tanks around my house, introduce you to some of my pets. In the meantime, folks, take care, enjoy the rest of the Global Science Show. See you soon.